Hello guys, in this video we'll be discussing what happens at the synapse. The synapse is simply the space between two neurons, and that's how neurons communicate, is by sending signals from neuron 1 to neuron 2. The signals basically go from electrical to chemical here at the synapse to electrical again at the postsynaptic neuron. So you're going to have basically these little circle things I'm going to draw here, which are called vesicles, that are going to store neurotransmitters that I'm drawing here just to represent, drawing these little dotted looking lines, whatever you want to call them. And they're going to be here at the terminal, right? And then you'll have something else here called a voltage-gated calcium channel. This channel is only going to open in the presence of a change in voltage, as the name implies. So once this electrical signal reaches this terminal, this channel is going to open and allow for the entry of calcium 2+, right? Which is calcium. Guys, I'm stupid. But so when calcium enters the cell, calcium is going to cause these vesicles to fuse with the plasma membrane and release their contents into the space. And these contents are your neurotransmitters, right? And you know your neurotransmitters, dopamine, norepinephrine, whatever, glutamate. And they have particular characteristics, like glutamate tends to act excitatory, but it also depends on the postsynaptic receptor, right? So I'm going to draw here my receptor, and I'm going to draw basically the neurotransmitter binding to it. And as a result, what's going to happen is, let's say if this was glutamate, the arrow is to represent basically what happens as a result of the binding, right? Sodium influx is going to occur. And what do you think happens when sodium enters a cell? Is it going to depolarize or hyperpolarize? Depolarizing means the cell becomes less negative, and hyperpolarized meaning it becomes more negative. And since sodium has a plus charge, it's going to depolarize the neuron. And if it reaches a threshold, it can generate another action potential. And that signal can be passed on again through this neuron to another neuron. But the idea here is you have an electrical to chemical event. You have these vesicles that store neurotransmitters, and the necessity of calcium for the release of these vesicles via the opening of the voltage-gated calcium channels. The neurotransmitters are going to be also sometimes referred to as just your ligand, which is just the molecule, the signaling molecule, binding to its receptor, right? Causing the opening of a channel. Let's say if this was glutamate, that will open a chloride channel. So if, let's say, not glutamate, sorry, if this was GABA, that would open a chloride channel, which will make the cell more negative, and sometimes that's referred to as hyperpolarized, as opposed to here, when we have that depolarizing event, you typically associate that with glutamate. But again, it always depends on your receptor type. Certain neurotransmitters can have different effects depending on the receptor type. So just to summarize again, you have an action potential that reaches the nerve terminal. That voltage-gated calcium channel opens. The influx of calcium causes these vesicles to fuse with the plasma membrane, which is this whole entire area right here, right? And once it fuses with the plasma membrane, it releases its contents, which are the neurotransmitters that bind to their receptor types, to their receptor, sorry. And those receptors are going to open and allow for ions to move in depending on which type of receptors they are. Basically, if let's say this is a glutamate receptor, bind to a glutamate receptor, it'll open a sodium channel, right? Which will cause the depolarizing of this neuron, and the signal will be basically propagated. But in the case of this, let's say we talk about chloride, it hyperpolarizes the neuron. it sends a negative signal. Obviously, there's not necessarily always an action potential. There's something called an EPSP or an IPSP. So signals can